All right. First of all, thank you for inviting me for this session. Like fast, five to like 3 to 5 years ago, I was also one of you guys, like students, uh, managing this MLA student society and doing what we like, what we love for the university and for our department. And I'm actually really grateful for to full to in to participate for this presentation to this session actually because uh, it's a really great thing to me. Okay, so as you all know, uh, I am Manuttara Gamage. I am Chem Informatics AI Systems Lead Engineering Scientist, Standard Seed Corporation. Um, Today I'm going to present about the role of AI in future of drug discovery and development. So in the content of this presentation, there are some important points to highlight: the basics of computational sciences and artificial intelligence, and the United States scientific ecosystems, because the company that I am currently working, it's based on US, from the Delaware state. So I'm currently working remotely. So there are a lot of things I need to say about this. I will discuss with the session within next slides. So building an AI in life science startup from scientific collaboration, vision for Sri Lanka to build scientific ecosystems, and introducing first open source drug discovery AI drug discovery platform, and the future of AI in medicine and healthcare, and final thoughts and call to action. Okay, this is the first things, and I am going to discuss computational sciences and the AI, the basic pillars of the things that actually exist in these fields. Okay, so this session is going to be some kind of a discussion thing, not I'm just presenting and doing the same thing again and again. I'll ask some questions from the audience, from the undergraduates. If you guys interested to call back, like to provide answers, it will be grateful to more deep dive, because this is all because like these all are science things. So it's really grateful, like if we can think logically and try to discuss things, not just listening and just hearing things. Okay, so from past years, from 90s, from 80s, from history of the humans, they always wanted to understand how proteins inside our human body, specifically humans, sorry, how proteins interact with more small molecules. What are small molecules? Small molecules is, you know, like small molecules are everywhere. Like in the foods you drink, in the for the foods you consume, in the beverages you drink, and in the laboratory you mix a lot of chemicals here and there, right? And do a lot of experiments, right? So small molecules, they are small molecules. Proteins are inside human body, in every cell, every tissue, right? So humans wanted to study how proteins will bind with small molecules, how the activation inhibition occurs, what diseases can be cured from that, what pathways are inhibiting or activating, what binding interactions are happening between a protein and a small molecule, what binding affinity, what are the toxicities, what are the solubility components, what are solubility of molecules with solvent. Okay, so to do that, from early days, also now, scientists used a lot of laboratory techniques, experimental methods. Anyone know about this machine that I put in here? Does anyone from the audience? Anyone know about that machine? Okay, so it's it's an NMR machine. So we call it nuclear magnetic resonance. So it's kind of a machine that actually identifies experimentally about the chemicals and proteins, how chemicals are interacting with proteins. That machine can give experimental values by kind of using a magnetic field, something stuff like that. Okay, so it's very popular in drug discovery and development technologies, so that's why I put it in there. So experimental methods, you can see here in this picture, people are using a lot of chemicals, a lot of laboratory equipments. But on the other hand, it will take a lot of money, right? A lot of money, a lot of time, a lot of laborers to do those laboratory experiments, right? So instead of doing these wet lab experiments at the laboratory, can we do it inside our computers? Can we do it? Did human actually did it? Yes, they did that. So actually, that thing that the laboratory, the thing that we can do inside computers, the experimental methods we can do will cost a lot of time and money, and also the labor cost will be reduced, right? So that's why actually humans wanted to use computers rather than just relying on the experimental methods. So it's kind of a shortcut to bypass. There's a lot of time, effort, and the cost to reduce it. That's why they are using computational method. So in computational methods, we use advanced computing algorithms, numerical methods, simulations, stuff like that, to identify, to do, to predict the things that we do in laboratory experiments. So these are few pictures I put on here to show how the computers visualize these things, the proteins and the small molecules, and how computers visualize the small molecules in like 2D diagrams and these stars charts and stuff like that. Okay, the computational models and stuff like that to emphasize that. So next slide. This is a very interesting story. Okay, um, does anyone watched this movie, The Imitation Game? Is anyone in this audience, youngsters, has anyone watched it? Like anyone have an idea? Does an idea about what that movie tells? Like anyone have? Okay, I will explain it little simply. Okay, so in 1940, during the Second World War, okay, Adolf Hitler used a machine called Enigma machine, right? Enigma machine to communicate with his commanders in his army, navy, air force, in his forces, to drop bombs in particular areas in Europe, like UK, France, the Germany all invaded those things, right? Those areas. So Hitler uses Enigma machine that can generate like 158 quanti trillion of possible combinations of messages to his commanders. Like it's changing, the message is changing time to time. It has possible combinations. So he was using that special message techniques to communicate with his commanders, because UK or France, the opponents can't understand the meaning of that message because it's a secret code, and always it's changing, right? It's really difficult to understand. He was using that Enigma machine to generate that secret code and to command his army. So by doing that, he destroyed a lot of enemy territories. Like he captured all the things, and he did a lot of bad things, right? We know that. And during that area, a scientist called Alan Turing came, and he used a machine called the bomb, a bomb machine, You can click this link and you can directly go to that description of that bomb machine. He created that bomb machine. Using that machine, he broke the code that was really impossible to solve by humans. Like if humans were there to solve it, but it will take like millions of years to break the Hitler's Enigma machine's code. May not millions, maybe 1,010,000 years, not millions. How it will take a lot of time, however, and it is really impossible to solve that secret code by ourselves using human mind. So Alan Turing used the bomb machine. He broke the code. He encrypted the secret code, secret messages that were delivering by Hitler to his commanders, and by using that, he actually support the UK, USA, and the opponents to end the war. Basically, they identified what Hitler was telling to his commanders, and they were placing the right army troops here and there. They finished the war by doing that actually. So you can see how. So this is what this yeah. This was a kind of a first computers that human ever developed. First type of a computer, the Bob machine, okay, to break the code of Enigma. So you can see how computers, the machines, can solve that complex problems that like to human will take to break in within a seconds. So you can see from this exact slide how it was done. And Alan Turing was the father of the modern computer science, and he did that. So it's a really great movie, The Imitation Game. Yeah, the guys and girls in here, definitely you need to watch that movie. So that's a really good example how these computers can solve complex problems. So to the next slide. Okay, in here, uh, when we use computers in biological research, like in life science research we call it chemistry and stuff, okay, to solve research problems, we need to use the computers, right? We need to give commands to the computer. We need to provide those structures, molecules, proteins to the computers. Without providing the structures, molecules, or proteins, how computers going to understand what we are going to tell? So we cannot directly tell computers to understand the things, right? We cannot directly give a protein to the computer and tell him, can you can you solve this problem, can you solve this problem? We can't do like that, okay? So computers cannot directly understand molecular structures. We can't directly understand. So in 1983, a scientist called David Weininger, he had his own company called Daylight Chemical Information System, stuff like that. He invented a language called Smiles. This is actually specific for informatics, okay? He innovated the language called Smiles. That thing revolutionized the field. Until now, we are using that chemical language. After like 30 to 40 years later, still we are using that because his innovation changed everything in the field. Okay, like I think Smiles means it's kind of a language, okay, chemical language. Like if you want to talk with another person in United States, their official language is English, right? So you need to speak in English to communicate with them. If you talk in Sinhala, they won't understand anything, right? So that's why you need English. You need English language to communicate with them. Likewise, you need Smiles to communicate with the computer. You can use Smiles to give molecular structures to the computer. So this is a Smiles notation that I put in here. The computer will convert this Smiles notation into binary digits, 1010, binary digits. And after doing it, It will understand how these miles can, like, how we gave the molecule to the computer, and it will give predictions based on that. That's how computers are understanding the molecular structures. SMILES is just a small examples. There are a lot of ways that we can provide those data, those molecule structures to computers using different type of languages. I will show in the next slide. Okay, this is a basic overview of SMILES. It's called Simplified Molecular Input Line Entry System. Um, like you can see here in SMILES, we kind of symbolize the names of the molecules, like the names of the molecules using some kind of a language. Okay, like for carbon we use C, for nitrogen we use N, for oxygen we use O, for sulfur we use S. Okay, for lower class letters we use it for aromatic ones, so upper class letter we use for aliphatic rings and stuff like that. And we use bonds, if it is a double bond it's two dashes, if it's a triple bond it's hash sign, and if it is a single bond like this one, a single dash. Sometimes we don't use a dash for single bonds. For branches we use parentheses, and broken labels, for like broken labels and numbers we use for rings. You can see here, in here, like we have carbon 1 2 3 4 5 6 cyclohexane, right? So if we use C1 in here, if we put number one here, it will create a ring, right? So 1 2 3 4 5, so five rings, 1 2 3 4 5 carbon atoms, and again we put a number on it, so it will come with the ring structure. Just like that, we use parentheses to indicate branches. So in here you can see, after these two carbons and after the third carbon, there is a parenthesis and there is a branch. Likewise, you can read about SMILES. It's kind of a bit complicated thing, but when you get used to it, it will be really easy. Okay, it will be really easy to use that. So like SMILES, there are other types of methods like languages, molecular languages we can use to provide data to the computer, like molecular graphs, connection tables, fingerprints. As the example of fingerprints, we can use Morgan fingerprints, PubChem fingerprints. As an example for connection tables, we can use like PDB IDs, a lot of PDB data. And molecular graphs are kind of graphs that have like a graph structure, so it can give some predictions. If we can combine it with a computer, stuff like that. Okay, so I can quickly go and show you stuff in here. So if you can download a file like a PDB file, you can see the things in there. I think you all can see my screen right now. So there are a lot of details related to the PDB file. PDB file means it is indicating this protein structure. This is a CB1 protein, human CB1 complex. Its agonist with this AM84 something like that ligand, and this is the human protein, and we can give that details like this to the computer, like this. This file is actually can be readable. This file can readable to the computer, so it will kind of visualize this stuff in those softwares. That's what it's basically doing. So if we go to PubChem database, you also can see different types of files you can download, and you can use that to give an input to the computers. Okay, All right. On the next slide, it's about computational sciences. Okay. So computational sciences are consist with different types of subjects. It's actually not a single thing. So you can see here, the basics of computational science are coming from mathematics, statistics, computer science. And on the other hand, biology, physics, and chemistry. Those are the basics. So computational sciences have branches. So these are the basics of computational sciences. Computational sciences build on these things like mathematics, stat, and CS, biology, physics, chemistry, and these are the branches of it. Okay, these are the branches. Like cheminformatics is one of the major branch in computational sciences. Bioinformatics is another major branch. Computational chemistry is another branch. Computational biology is another branch. Neuroinformatics, healthcare informatics, computational physics. There are a lot of stuff, lot of branches in computational sciences. It's all about combining computer science with the basics of biology, physics, and chemistry with a small amount of statistics, not small, uh, with the amount of statistics and mathematics. Okay, so uh, so informatics subjects. So what are the informatics subjects? Cheminformatics, bioinformatics. Why we use informatics name onto it? Neuroinformatics, informatics. What's it? What is that? So if I get an example for cheminformatics, then you can. I will show you a data set of it for an example. You can see this is one of the company's data sheet. So you can see there are a lot of data on it, right? Lot of data. PDBIDs, protein families, primary target, primary diseases, stuff like that. There are millions of billions of data in this sheet, and as humans, like it is really difficult us to go through this every point, every single name, right? It's too complex, like billions of trillions of data in it, right? So that's where actually the computers came, like come in, and what they do is like they help to get predictions using that large sets of data. Like in cheminformatics, we use large set of chemical data, right? These are chemical data. It consists of a lot of molecules, lot of protein targets, right? So those are chemical data. In bioinformatics, most of the time, we are using a lot of genomics data, genomes, adenine, guanine, thymine, cytosine. I can show a few examples. Like if you go to this data set, uh, you can see this is genomics data. There are millions of genomic data in here, like a lot of letters, right? Can you, like humans, can you kind of understand these things? Can you give predictions just looking at this and tell? You can't do that. You can't do that. It's really difficult. That's why we need computers. So in cheminformatics, bioinformatics, in informatics related subjects, we basically use computer science, mathematics, statistics with tons of biological data. Like in bioinformatics, in cheminformatics, yeah. In neuroinformatics, a lot of data into neuroscience. In cheminformatics, a lot of data into chemical sciences, chemistry. In bioinformatics, a lot of genomics data. Likewise. So that's how we can distinguish these subjects, right? So in computational chemistry and computational biology, the things are a bit different. In computational chemistry, actually we use a lot of softwares, a lot of tools. to identify how proteins interact with small molecules, like in this picture. This is like a small pharmacophore model. Okay, you can see the hydrogen donor acceptor or stuff like that in here. We use that pharmacophore model to identify how proteins bind with small molecules, how many interactions it will cause, like it will create, how many, what are the type of interactions, how protein to protein interacts, how protein to small molecule interacts, how protein to polymer interacts, how polymer to small molecule interacts, something stuff like that. In computational biology, we use biological systems like membranes, large protein complexes. It's a bit similar to computational chemistry stuff, but they have big kind of differences. But yeah, so likewise, this is how we can categorize computational sciences, and this is what actually I know about these things. And uh, so in computational chemistry and computational biology, actually we actually don't use lot of computational algorithms. We don't use a lot of machine learning or deep learning algorithms. We just using a software to analyze how these molecules behave. But in cheminformatics, bioinformatics, stuff like that, we have a lot of, like, we use a lot of uh, AI models, machine learning models, to interpret things from the large data sets. So that's the difference. I think you can understand the things I am telling in here. So if you have any question, any doubt, please ask me questions and I will give the answers uh, I can, right? So I told you, like, cheminformatics in bioinformatics. We are using a lot of AI and ML stuff because we can't analyze that tons of biological and chemical data just with our human brains. We need computers, we need machines to solve them, we need machines to give predictions using that. So AI actually, I know it's a very popular thing these days because you can use an AI model with your phone, right? You have ChatGPT or Gemini AI. You can just switch it to the model that you want to use, and you can ask questions and you can get predictions. It's really popular in 2026, but actually it was rooted. The history of AI goes back to 90s, more than that. Okay, so the Turing test was one of the significant tests that was done by Alan Turing, the same scientist I described in here. He was the father of modern computer science. So he actually did the test, like he used human and a computer and asked the same question from the both. And from that test, however, he identified the computers also can give human like answers. And after like 50 years or later, or later after that, in 2026, we can identify that AI can actually behave like humans. It can talk like humans. It can give predictions like humans. So actually, it was beginning in 1950 and in 90s. So ELISA is one of the main innovation at that time, and in these days also for the chatbot, the ELISA technique is actually using. So you can see the whole pipeline, all the timeline in here from 1642 to 2018. In 2018, a lot of universities start university courses related to that, related to AI. And adversarial neural networks invented in 2014. And the history of this is massive. I can't explain the history within a small one slide, so you guys need to go and read about it and just setting the overview. Okay, so traditional modeling means traditional computer algorithms and machine learning. What is the difference of that? So in traditional things, right, traditional algorithms, we use our own handcrafted model with the data that we want to analyze, and we get results what we want using the computer. Okay, that's a traditional method. But in machine learning, the things get bit different. Not bit, actually very different. Like first we use a sample data, and also we use accepted results. The accepted results mean the results that actually we want from, right? So we use computer to give that data to it, and then we develop another model. We develop a model using that sample data and the experimental results. So with the expected results, right? A new model. Then we use that build model, the trained model actually. We call it trained model. I'm just explaining this really simply. We use that trained model, the model we developed in here. We use that model and we give new data to it. New data. New means we will ask questions from it. Like if we I know you are asking questions from chat gpt, what is the climate today, right? That is a new data, right? We can ask from the model new questions from it, right? Then the model will provide new results, new predictions. That's the difference of traditional one and the machine learning one. Machine learning, like, we can build our own model by giving the expected results, and we can get predictions. Okay, the small example of it. I can go really deep into it. I will go step by step. Okay, in here, um, I will explain about AI, ML, deep learning. Like, what is the terms? What is the meaning of these names? Okay, the AI is actually enable machine to mimic human behavior, artificial intelligence. You know, everyone knows it. Okay, so machine learning is a subset of AI that we like use statistical methods to enable machine to improve with experience. Okay, so in deep learning we use like multi-neural networks to get predictions. Okay, so in machine learning actually, we as a human, as a scientist, we kind of process the data that we need to give to the model. We extract feature extraction done by us. But in deep learning, you can see here, deep learning, feature extraction and the classification both done by the model. So it is fully autonomous. Deep learning is fully autonomous. Okay, so you can see the differences. Focuses on feature extraction, requires more manual intervention, works well in the structured data. Deep learning, it performs automatic feature extractions, no need manual interventions. It requires large amount of data, and it excels at processing unstructured data as well. Okay, so we can provide unstructured data to it as well, so it can, like, it has the ability to create, to provide the predictions autonomously. So neural networks are the basics of these deep learning things, deep learning models. Okay, so in the artificial intelligence, neural networks are the basics. Okay, so neural network are the foundation of it. Using neural networks, we kind of train models, retrain models, and we get predictions. That's all. So neural networks. So from early days, human, like I said earlier, human always wanted to study how these neurons, how the neurons in our human brains behaves, how they actually work. Can we mimic that? Can we build an artificial neural network? Can we build an artificial one? using programming, using computers. And they did that. They created the neural network called artificial neural networks based on the biological neural network. The biological neural network are not equal to artificial neural networks. The structures is totally different because it's mathematics and statistics, something stuff like that in AI. But they were trying to mimic the things that can be done by humans, right? By developing AI, right? So this is the architecture of a neural network. Most of the AI, most of the neural networks has three layers: input, hidden layers, and output. So data can be given through the input layer, so it will move through the hidden layers, and each layer perform matrix operations using weights and biases. And there's activation functions like that shapes signal before passing it. Means like the signal is good to go, it will say 1, so signal can go. If the signal is not good to go, it will say 0, so signal won't go, something stuff like that. And output layer gives the final predictions. And a back propagation is something like the model can use the model can kind of adjust its errors by kind of doing some mathematical calculations. It can solve its errors. Okay, that's back propagation. Okay, so this is a basics of how biological neural networks and how artificial neural networks behaves. Okay, uh, so there are a lot of artificial neural network types actually. Okay, this is there are a lot of types. Like there are more than 20, I think, lot of types. Recurrent neural networks, long short-term memory networks. It's adaptation of RNN as well. Okay, deep convolutional neural networks, deep belief neural network, feed forward neural network, graph neural networks. There are a lot of neural network types. So people using it for different purposes. Like convolutional neural networks, they used for like image identification, scanning. LSTMs they use for drug discovery and something stuff like that. Basically, for the drug discovery, they use LSTMs. For molecule generation, uh, what they do is actually they use these models. They train them. They train them by giving those data, like this miles data. This data they can train the models by giving the data that they want. Okay, so like I said you earlier, they have kind of different layers. Some adaptations came later on. So deep feed forward neural network is one of the basic neural networks. After that, a lot of new type of neural networks like recurrent neural networks came, and their structures are a bit different from the oldest ones. They have memory cells, like LSTM have memory cells. So there are differences. Like convolutional have convolution pools. There are differences. Like they adapted with time. Okay. So in cheminformatics, people use different tools, different AI tools like reinvent. In reinvent, they developed it using recurrent neural networks. In Decima, they use some kind of novel method of convolutional neural networks. In Chemprop, they use graph neural network. Okay, so in different tools, they have different neural network methods, neural network types. But however, they used kind of these data to train this model and to get the predictions. Okay, all right, then to the next section. So how US scientific communities evolve into startups and to major tech ventures. So before going to that into deep, I need to introduce you to this open source software and the closed sourced proprietary softwares. Proprietary softwares means closed source ones. Open source softwares means is open for all. Like um, open source softwares mean you can have the source code of that open source software from a public repository. Like if you think the internet, you can, if you think Google, you can just type it and find the source code of the open software, open source of the source code of open source software in the web. You can find it really easily. But in closed source software, you can't find the source code. Source code means the code that was used to develop the software. These softwares were developed using coding, using programming languages, Python, HTML, Java, C++, CSS, something like that. Okay. So closed source softwares basically we can't we don't have the access for the source code. For open source software, we have the access. And also, open source softwares are free to use and they are highly customizable. They are community driven. Okay, community driven. So they are third party. So they are transparent. Time to market can take some time. Will take some time because it's community driven. No vendor lock. It's really flexible. Availability is long time. Okay. So in other hand, closed source software, they are restricted to access. Cost, there is always a cost. You need to pay subscription to buy a closed source software, right? Customization is really limited because you don't have the access to the source code. Without having the access to the source code, you can't customize the software, right? The support, you need to get the support from the company, right? You can't get the support from the community. You always need to contact the company to get the support, right? In the laboratories, in medical laboratories, people use a lot of machines and they have a lot of softwares. Like what happen if the software breaks in some kind of a machine? You need to talk to the machine supplier, right? You need to talk to them and you need to get the machine from the get support from it, right? So you need to get support from the vendor. Just like so open source software examples: Android, VLC player, a lot of browsers, like if you can know here, the Firefox. Okay. And Linux is one of the most popular open source software, as in free to use. Anyone can download and use it. On the other hand, closed source softwares, Photoshop, you can't use it without buying a license. So what we do is we crack the software and use it, right? We crack it. So it's not a good thing, but we do it. So Microsoft, their softwares also, people crack it and use cracked versions, and some virus guards, people use cracked versions of it because we can't afford the cost. So they are not freely available. Microsoft Office also like that. But in open source culture, open source softwares, they have softwares that everyone can free to use. And also source codes are free to accessible for everyone. Really easy. Okay. So in 2022, this is my career pathway that I came from 2022 and beyond that. I did my undergraduate research in drug discovery, nano drug discovery actually. Then in 2023, I learned coding, Python, bioinformatics, and later on, I joined Global Chem in 2023. Like I said earlier, Global Chem is open source government. Government means it's a community focused on transforming common chemical names into 1D molecular representations, facilitating universal recording and government. So I joined it in 2023. So what opens what globe what is Global Chem actually doing? Okay, So in global chem, it was a community driven open source governance, a software system, actually a software system, but a collaboration of the people. And my supervisor was the leader of that community. He was doing a lot of work in there. And what actually we did in that open source project was we contacted the different industries all around the world, like food industry, education, environmental industry, material science, narcotic, warfare, proteins, organic synthesis, interstellar space exploration organizations. Okay, lot of people we contacted, and we told them, tell what are the chemicals that you are using in your specific industries. Like we asked from food science people, a scientist working in a food laboratory, we asked from him, what are the chemicals that you are using to process fruit, like to create foods. Okay, they use color additives, something stuff like that, salts. In medicinal chemistry, they use a lot of chemicals, right? In in warfare, people use some organic phosphorus, nerve agents. So those are the chemicals in different industries. We told people to gather all those chemicals from different industries. We told them to gather and put into our global chemistry's knowledge graph. That's what we did. So people came in, a lot of collaborators. It's actually more than 100. It's actually a lot of people. Okay, a lot of people came, and they provided their chemical names, and they categorized them, and into several things, like plants or something like that, or color additives, something like that. We created smile representation for each chemical, and we with the categorization, and we put into the knowledge graph. Okay, so this is a small brief example of that knowledge graph. So we put that knowledge graph as a Python package. So everyone in the world, scientists, researchers, knowledge seekers, can download this Python package from us, so they can use it for their own purposes. Specifically for cheminformatics research, this knowledge graph was really useful for the people. So if you click that button, this link, you can directly go and see the Python package. You can download it. There were plenty of collaborators for this project all over the world. um, so yeah, also if you can click this in here, you can see people are downloading it daily. Okay, people are downloading this is the analytics of the software package. It's have more than, I think, more than 2 lakhs of downloads. Not 2 lakhs, like yeah, around 2 lakhs of download, like 1 lakh and 77,000 of downloads. You can see it here. People are downloading it all over the world at this time. Also, you can see there are different versions of this software we are releasing time to time. I worked as a development operations engineer in this workflow. I release this software time to time. So a lot of people was using this software, downloading. As you can see here, the peak was in December 26th. There were like more than 20 downloads, I think. And also March 18th, there were like 4, 5, 6, 7, 8, 9, more than 10 downloads. Okay, people are downloading it every time all around the world. So it's really useful, and so that we created in here. Um, more than 10 downloads per day, more than 1 lakh and 77,000 downloads overall, and a lot of collaborators. Okay. um, I think you guys can understood the things I described until now. So I'm going to go to the next slide. Okay. This one is called Mother Nature AI. Okay. Mother Nature AI was built by standards, no, built by Global Community. I was one of the developer of this project as well. We actually did was we used reinvent a recurrent neural network. It's a type of neural network. Reinvent is actually a software that was released by AstraZeneca. It based on RNN, recurrent neural networks. Yeah, and we use that to generate new molecules. Like we have a lot of data that we gathered from this knowledge graph by gathering the knowledge, by gathering the data from the people, and we use that data uh, to generate novel molecules. Okay, novel molecules in specific industries, in food industry, cannabis, medical chemistry, sports, something like that. Okay, what we did was first user comes in and he will ask our AI system, our Mother Nature AI from ask Mother Nature AI from to generate chemicals based on this category. All right, so it will create a policy. This per year section will create a policy. This is a layer. Okay, this is a layer. It creates a policy based on the inputs that was given by the person. The policy then will create novel molecule. The agent, the sorry, the policy will be handed over to the agent, and the agent adopts that policy and then generate new molecules based on it. That was actually doing in this Mother Nature AI system. Okay, this is also an open source one, so everyone can use it. And these are the things actually we did in global. Okay, so later on, global became like it became a startup company later on. So from this section, I talked about how to build, how we were building a startup from a scientific collaboration that we get from Global Game. Okay, and we got from Global Game. Right. So if anyone has any questions, please feel free to ask. Okay. So in 2023, I joined Global Game. Then I became open source research scientist, and I did a lot of DevOps work in there. I released software packages, and 2024 I became a community manager, and 2024 also I joined the standard seed build at that time with the community's help. It's a startup company based on the community. It's a corporation, so I joined company as an intern. Then I became a junior informatician. Later on, with the experience, I became the lead. Okay, so in here, right? In here, just need to take this below. In here, you can see we gathered a lot of people from the community, like plant cultivators, genomic companies, biotechnology companies, wellness product development companies, beverage companies, farmers, NGOs. Those communities, like those people, were combined into global chemistry's community. Okay, so In standard seed, what we did was, it's actually a novel thing. Uh, it's actually validating things, okay, using AI. I will describe it. So think this thing, this the thing in here as a beverage. I think it is a beverage from a beverage company from the community. In a beverage there are chemicals. If we get a chemical plant-based beverage, there are a lot of chemicals on it, right? Hundred and thousand chemicals. Standard seed can identify those chemicals. Like if the user can provide the chemicals, or a paper or something like that, it will be really useful. Like if you have the chemicals of that chemical of the this beverage, we have that our own AI system that can identify what protein targets will bind with the chemicals of this beverage. What protein targets in human body can bind with these chemicals, the chemicals in the beverages, in the beverage product. So people had lot of components, like not also the beverages. People had a lot of supplementary products, hair oils, beverages, food meat drinks. Those are beverages, no. And some companies, they provided chemicals they want to analyze. So our AI system can identify which chemical can bind with which protein targets inside the human body. Inside human body there are millions of targets. So our AI system can identify those millions of targets. So it can identify what protein will bind with what chemical from this beverage. So we can validate this beverage, or validate this hair oil, or validate this wellness product by using our own artificial intelligence system. Okay, that's what standard seed are doing actually. Okay, so in here we basically use lock and key model, where chemical is the key and the proteins, the binding site is the lock. Okay, there are locks, the binding sites. There are millions of binding sites in human body. There are millions of proteins, also millions of binding sites. Our small molecule, sorry, our AI system can identify, like I said earlier, which protein, which protein will bind with which chemical and to which receptor, and how much binding affinity it will make, and how the mechanism of actions, how the protein pathways activates, like how many diseases, so what are the diseases can be cured or something type like that, and what are the cellular and subcellular targets. uh, we have our AI system that can predict all those things. Okay, so this is the AI system, standard seed botanical artificial intelligence factory. This is called actually the project Rebecca. I am actually leading this project right now at the company. So in this project, like I have said you earlier, we have different type of AI agents. Okay, AI agents. We have agent for molecular docking. We have agent for docking reasoning. We have agent for molecular dynamic simulation. We have agent for toxicity prediction. We have agent for human protein target detection. We have agent for dissolving things to identify the solubility. Okay, we have agent for mass spectrometry. So we combine those we agents into a one single platform. That platform is called Rebecca, and it is called SSC, Botanical Artificial Intelligence Factory. So a lot of people in the wellness product gave us a lot of botanicals, like a lot of plants actually, because they are creating their beverages and their hair is using plant material. So they gave us a lot of chemical components they want to analyze through the system. So that's why we named this as Botanical AI Intelligence Factory. But the name was changed to Rebecca actually. So we are using different of methodologies, different of AI tools, a lot of uh, tools, software tools to develop this agentic AI system. Okay. Um, so the AI models, and there's a difference between actually an AI and an AI model. Okay. So sorry. The difference between AI model and the agentic system. I was mistake. Uh, AI models is the theory, actually the theory behind these things. AI models actually doing the predictions behind these tools. Okay, the agentic system built upon these all the models. So that's the difference between. So this is agentic system. This is this will do the customer wanted. Like if you can click that link, this link, you can see the reports that we are providing for the customer. You can see the customer is providing a chemical. We are analyzing it through our agentic AI system, and we are providing protein data, protein types. What are the protein types that this chemical will bind, and also the toxicity of the chemical. We will provide this, and also the solubility and the mass spectrometry. There's all were analyzed through our agentic system. Cellular locations, tissue expressions. So this is a lot of data. Okay, that was the data actually generated using this system. Okay, so Becky AI is one of another agentic AI system that actually using in our company. So bioinformatics, cheminformatics, related tools are actually belong to Project Rebecca. This project. So I am only working in this specific area of the huge agentic AI system. So our agentic AI systems have neuroinformatics, clinical intels, formulation, stuff like that. So it's really huge. And I'm just working on this small section only for now. So yeah. So this is our company's. Um,
Excuse me, Mr. Anutra, there is a question.
okay, what's it?
Is there any model that can be used to identify a specific peptide binder from a library?
Yeah, there are models that you can use to identify that. I think you are asking from the company's perspective. I think I will read the
Thank you.
question. That can be used for identification of specific peptide binder from the library. Yeah, peptide binder. Yeah,
Can you hear me, Mr. Anuttar? Is it
yeah, yeah.
okay to?
Yeah,
Okay,
no
so
problem.
thank you very much for your nice presentation. What
Okay.
my question meant was,
Yeah,
uh, when we consider about different protein
yeah.
targets, when we consider different receptors in our body,
Hmm.
so there are specific points we are targeting
Hmm.
when we consider about different biological pathways.
Yes.
So in that case, for example, in case of cancer therapeutics,
Yes. Madam, actually, yeah. I can't hear you actually. Madam, for the answer for your question, actually, our AI system can identify what um like like what specific residues will bind with the small molecules. And actually the difference like the distance between the small molecules and the peptide and the bonds that the non-covalent interactions, hydrogen bonds, those things, our AI model can identify those things. Madam. Okay, okay. Like, actually, madam, like X, Y, Z coordinates also can be identified. Where the pocket is, where the binding pocket, and what interaction it makes. Lot of data, yeah. So okay, this is seed to medical market. This is the whole agentic pipeline, whole research pipeline of our company. So if someone wanted us to develop, like someone wanted to collaborate with us to develop a to develop a medical drug, a novel drug. This is the way you can do it. So basically, you can provide what chemicals you want to analyze, and you can give it to us. We will do all the analysis from zero to from basic level to the top level. Okay. Uh, think that you give if some person from the pharmaceutical industry or some beverage company provides us like 100 chemicals from a plant. We can use that chemical components through our AI system. This is where I actually work on this little system. That AI system, our AI system can identify, like I said, all the properties, all the binding targets, all the proteins, all the solubility, all the data. And we can use that data to do preclinical studies, cancer cell lining screening, clinical trials, safety and toxicity assessment, and NDI, OTC, IND, NDA, and finally we can help that that the provider who provided us the chemical is to put a medical, like put a drug, a novel drug to cure something, a doctor ready drug to cure some disease to the market. We can help to do all the things using our this platform. So AI analysis is only a single component of this whole pipeline. I'm just working in this single section only. Okay. So this is the company. My supervisor is Dr. Suleiman Sharif. He's the founder of Standard Seed Corporation, and Mike Foster is from Linux Foundation. He's the CEO of this company. We currently have 17 partner organizations, specifically in US, also in some, also in UK as well. We are seeking for more partnerships, more scientific collaboratives, and this is Damiola Burden. He's one of the cheminformatics advisors from earlier days, also for me, and I am the lead engineering scientist for that specific cheminformatics AI systems. And Kalana is currently working as a junior cheminformatician. Shakir also working as a junior cheminformatician. And Pamaka is the management assistant. He's also from the MLS department. Uh, is one of my batchmates, and David is in sales and educations. We have a lot of partner organizations, so our structure is lot more complex than this. Sorry. So actually, I just overview this specifically to understand it easily. Okay, so these are our partners: MCR Labs, Melo Fellow, Arvida Labs, um, CCDC from England, Canometrics from US, lot of partners. Okay, these are publications and innovations actually we did past few years. Um, we published those in a few journals as well, and MDPA elsewhere, and we published them in those journals. And you can see this is actually related to deep learning, LSTM. uh, I will explain that project really easily, so you can understand, because this project is a really good thing for people who want to learn how these uh, AI models can use to innovate novel drugs. Okay. um, so EG5, is a mitotic kinesine. It's a motor protein essential for forming bipolar mitotic spindle duration cell deviation. Okay, you can see here EG5. Uh, it is helpful for in cell deviation. So it's overexpressed specifically in cancers like breast, pancreatic, gastric. So it's a prime target for antimitotic therapy. Okay, so what we did was, we use neural networks, like I said earlier, AI models, to generate novel molecules. To generate novel molecules, and then we did some identification through the through our system, and we identified the generated novel molecules were really great, really good to cure cancers. Okay, to inhibit these proteins. Okay, so in here, there are two type of neural networks actually. Okay, this is a forward neural network. This one is a recurrent neural network. A forward neural network is just a simple one, right? Okay, a person can provide inputs, and there is a hidden layer, and there is output layer. So the input will come from here and go through this layer, and output will produce. So in recurrent neural networks, the output layer feedbacks, like it will provide the data that is going through this back to the hidden layer, and it will kind of enhances the output. Like it's like relearning something. Okay, it's a relearning something. So for this project, we never used forward neural network. We use recurrent neural network because it has the learning, like uh, providing its own output to its as its input to like reduce its own mistakes. That's why you use recurrent neural network for this project. So um, but recurrent neural networks, RNNs, have some issues. Okay, when the messages, the inputs passing through these hidden layers, the hidden layer. Okay, the sensitivity of the message will get reduced time to time. Okay, um, thank you. With reduced time to time. But in LSTM networks, because of the special gates, the the sensitivity of the input that we provided won't shade with the time. So that's why we use LSTM network to this research. Okay. So like I said you earlier, LSTM research, LSTM models have specific cells and gates, like cell state, input gate, forget gate, output gate. Okay. When you provide input through the input gate, there's a special thing called forget gate, and it kind of accept or reject, do the function like that. So if the forget gate want to retain the input that we provided, it will say yes, it will say 1, and it will pass through output gate. If the forget gate do not want to retain the thing that we provided as the input, it will forget it. So likewise, we can we can train the model again and again and again by providing the output as the input again and again. It won't shed with the time. That why that was the reason we was using LSTM for this project. Okay, so these are the small gates and the cell states. Okay. So this is what we actually did in here. We selected 1.5 million compounds from Campbell database. Then we prepared those molecules using several methods like several tools, RDKit and OpenBabel. Then we trained and retrained using LSTM data, so using LSTM models by providing these molecules. We trained the model in here. And using this model, we provided outputs. We generated outputs. We generated novel molecules. Okay, novel molecules. Then we did chemical space analysis, virtual screening, structural based screening to identify the top molecules, and we did ADMET prediction, and finally estimated the binding energy using docking study. And also, as a final step, we did simulations and identified the molecules that can cure cancers, that can bind this the receptor and the molecules that can inhibit this protein. Okay, so actually we developed that using artificial intelligence. We generated the molecules from the LSTM model. Okay, like I have said you earlier, the problem that we are getting in standard neural network is vanishing gradient problem. I forgot it to tell you. So in here in LSTM networks, uh, we can we don't have that problem because we have these gates in every each cell in hidden layer. So gate will bypass that problem, the vanishing gradient problem. So standard seed have upcoming novel innovations, novel toxicity and solubility prediction tool. Already we developed an open source tool called TrackMyPDB, a novel protein identification tool will release soon. The new publications as new publication, and also we are currently focusing on anti-skin development drugs, uh, anti-skin cancer drugs. Uh, Actually, it's currently in preclinical stage, and soon I think it can be move into the other phases, like the other phases in the pipeline. And next thing, vision for Sri Lanka to build a scientific ecosystem from ground up. Um, in Sri Lanka we have a lot of cricket communities, a lot of cricket players, right? So every child takes a bat, take the ball, go to the ground. In evenings they play cricket, and later on they become gold cricket, like global cricket players. And we have that type of a community-driven cricket culture in Sri Lanka. But actually, for scientific things, we actually don't have that type of a scientific community or culture, right? So so it is really difficult for us to capture the portion of the global market because we actually don't have from our roots. So we have cricket from our roots, but science actually we need to come from our roots. Nowadays people are trying to build those things, but actually really uh, like very little people actually doing this. Like we don't have much scientific communities in Sri Lanka. So in US the story is different. They have open source communities everywhere. They are using a lot of platforms like Reddit, LinkedIn. They have communities, so the PhD students, a lot of people are gathering into one platform. The business people, and they are talking each other, developing new projects. We don't have that culture. Actually, we need that culture in Sri Lanka, I think, to capture some portion of the scientific market from the global. So the lack of scientific, we have challenges: lack of scientific communities, lack of scientific ideas, and the funding is also less. The innovations are also limited to papers. A lot of most of the scientific time in Sri Lanka. So actually, we need to go beyond that. We need to go beyond research papers to value-added product and to the service. Okay, the real world actually not always the research papers, the skills, the leadership also a very important task, like very important thing. So you can see in here the neighbor country India is kind of leading in startup ventures because they are going beyond the research publications, and we are going beyond um, the basic things of science. So that's why they are kind of passing all the nations India. They are capturing a great portion of the world by doing these ventures. Okay. So as myself, to build some kind of a science, I always wanted to build some kind of scientific community for Sri Lanka, scientific community for Sri Lanka. So that's why I built this. It's a small institute and a scientific community, so everyone can join to it, everyone can collaborate to this. um, and from this community you can learn how to think out of the box, to be innovative, to contribute to science, how to contribute to science, to be a leader. And there are a few open source projects as well in here. I developed for the students to learn how to contribute to open source projects, because actually we don't have that much of scientific open source projects in Sri Lanka. So I actually built this for students. So some students, like some junior students, came with me and helped me to build this project, and they got their opportunities in several places like the standard seed. So that was one of the thing and I did to avoid this problem, the lack of scientific communities problem in Sri Lanka. So I will little bit explain. There are only few slides to complete. I will little bit explain about the project that I actually did in ESI. So everyone can contribute for this project. In every undergraduate, every any person. Okay, once my supervisor asked from me how to chemically categorize the ancient Ayurvedic traditional medicinal formulations. You know that in Ayurvedic products there are a lot of chemicals, lot of subcomponents. So actually, it was really difficult to categorize them because of this, because of the complexity of the components in the Ayurvedic formulations. So I developed this open source AI drug discovery platform called Traditional Chemistry. So it has two components. One is it has product names of Ayurvedic formulations and benefits, diseases, chemical composition, and smiles notation. That's language that can understand by computers. So I created a separate database from it. Okay. So this one is a chatbot. So we can LLM, large language model. So if you can connect that database to the large language model, people can easily use natural language. Like people can type and ask things from the chatbot about the traditional, like about the molecules in traditional chemical data, right? So what I did was, actually we did was, I had a few juniors who worked with this project. We analyzed the text from ancient textbooks. We analyzed the text and we identified the formulation. We chemically categorized it, and we identified the chemical components of each formulation, and we created a database and created a chemical engine based on it. Okay, so this chemical engine is connected to the each node, so users can use this LLM to get predictions. Okay. The software architecture of this project is bit complicated, but I will tell it in briefly. So if you think one formulation from an Ayurvedic component, there are multiple plants and multiple components, right? Plant A, B, C, and component A, B, C. So if we think one plant, it will consist of 500 chemicals. If we consider plant B, it will have 400 chemicals. If we consider plant C, it will have 200 chemicals. So the chemicals was categorized and provided and stored in smiles format, so the computers can understand easily. And those data also consisted with those diseases, and also with these things like product name, benefit, diseases, and something and stuff like that. And we stored in the database, and I use LangChain and connected that database with a large language model, so user can prompt in their screen and get predictions from this database. Okay, so I can show a small demonstration of it if you guys want to see it. uh, Anyone can ask questions from this. You need to use your open like your own API key for this, like OpenAI or Gemini AI, or you need to use your own API key to use this. You just need to ask questions from it. It will provide answers. Where if you say hi, so it has this data. Hello, hi, how can I assist today? With information on traditional chemistry or chemistry. Okay. It consists of these databases, consists of these medicines. So anyone can chat with this and ask questions. What chemicals in this one? Okay. It will provide answers. So more the data we provided to this database, so anyone can contribute to this project. Okay, this is open source project. Anyone can add data to this project. So this database will be expanded, so the output from this chatbot will enhance with time to time. If you go to this repository, you can see all the data that I was gathering here. So anyone who wanted to contribute to this project just need to put this smile string, just need to identify and categorize already component from the ancient textbooks. Okay, by doing that, anyone can contribute to this project, and that's what I am doing here. uh, so it will be really good for the people, for the scientific community. Okay, so I'm going to the final sections of this presentation. So the future of artificial intelligence in medicine and healthcare. So in healthcare and in medicine, AI used in several type of things like medical imaging and diagnostic, clinical support, clinical decision making, predictive analysis, robot assisted surgery, virtual assistant and chatbots, drug discovery and development, healthcare administrative operations, lot of uses. Okay, I work only this specific area. So there are a lot of areas that AI is used. So if you have the knowledge, if you can enhance the knowledge related to artificial intelligence, you can go and study, like you can go and work in these areas and do innovations on it. Okay. So these are the applications in of AI applications in healthcare. Like I said you earlier, you can read these things. It's publicly available in every place. You can read and understand. So this I already explained about this earlier. So traditional methods like wet lab methods will take 10 to 15 years to develop a drug, and cost will be very huge, like 2 billion of dollars. Okay, a lot of money. By using accelerated methods, we can reduce the cost by 70%, and also time will be more quicker. We can develop a drug within like 3 to 6 years. We can do it more than 3 to 6 years actually these days because of the agentic AI system, like I showed earlier in our company's AI structure. Oh, this is the applications of AI in drug discovery, target validation, drug screening and lead discovery, drug optimization and design, preclinical and clinical development. I actually showed these things earlier inside our company's course. We are doing these all things inside our agentic AI system, and we are currently actually we are developing it time to time, updating it with the customer data. So global chem and this community ISI is open for everyone. If someone wanted to learn about open source contribution and someone want to contribute to open source project, please join for these communities. If you click these links, you can directly join to the communities. Global chem is actually belongs to my supervisor, but now I am handling it now. And ISI is one of the own scientific communities of mine and of my few students who helped me to develop. It's an open source thing, so everyone can learn things from it. For at Standard Seed, we are actually doing a lot of collaborative research all over the world and all over the universities, especially my supervisor, Sul Sharif. He's in MIT, so he's kind of doing neuroinformatics research. Uh, he's in McGraw Institute of Brain Research as MIT, so he's actually doing neuroinformatics related research, and he's combining the neuroinformatics with the standard seeds AI agentic system. So that's what he's doing, and also currently we have kind of I started anti diabetic plant based drug discovery system, drug discovery research with Slit University, and we are kind of companies kind of making some arrangements with the Slit. And at Faculty of Medicine, I kind of do some personal projects related to nanomedicine in there also. And our company is open for academic partnerships always. The cost is free. So if some university or some department wants to collaborate with us for AI uh, artificial intelligence driven scientific research, we are always open to it. There will be some agreements you need to make before we kind of collaborating with each other, but it's for free for now. My supervisor actually told me to tell this to the community in Sri Lanka. So it will be really good to make some scientific collaborations to go beyond research papers to the medical market to serve value-added service or a product. Uh, so from this link, you can go to Standard Seed website. It includes all the details I told you. Yeah, global partnerships in here, partnership section, and uh, yeah, all the details are in here. To anyone who want to partner with us with Standard Seed, can just contact my supervisor or just send me a message or an email, me or to Pamalka also you can send. If it will be better if you can directly contact my supervisor because he's in charge of everything. Um, so the thing we know is a drop. What we don't know is an ocean. This picture I actually got from one of the famous TV series of all time, dark TV series, television series. So the thing is actually right. There are a lot of things I also need to learn, and this is all about the presentation for today. And thank you very much for giving me this opportunity again. And if you have any questions or anything, ask from me, and I will provide answers whatever I can. And if universities and departments of any person, a company, something like that, want to collaborate with Standard Seed, we are open always. So you can use these contact details and contact us. So thank you very much for giving me this opportunity. So I think it's time me to now to wind up the session. Thank you very much. Thank you.
Thank you very much, sir, for allocating your valuable time and sharing your insightful knowledge with us. We truly appreciate your presence and the inspiration you have given us today. We now open the floor for questions. This is your opportunity to engage directly with our esteemed speaker. Please feel free to unmute your microphones and ask your questions, or you may submit them via the chat box. Questions are welcome in both English and Sinhala.
There's a question raised for you in the chat box.
Yeah, I'm looking. As ML student, we don't know much about AI ML, how we do develop knowledge, and what are the opportunities we can get. There are plenty of opportunities you can get, and to learn AI and ML stuff, what I recommend is to do a self study, like not doing a lot of courses and stuff like that. It is better you can do self studies. Of course you will need some guidance from a senior person, but there are plenty of resources out there, a lot of research articles out there. You can go and just read and identify a lot of things related to AI. It's a lot of resources are free actually. A lot of videos in the YouTube also you can you can learn from them as well. And opportunities, uh, AI ML in life science opportunities, it's very limited in Sri Lanka. But outside Sri Lanka, there are plenty of opportunities. Actually, you can even apply from here to those companies as well, because like informatics research things are a lot of remote work. It's done through the computer. So if you can enhance the knowledge of yours in AI ML things, and also if we can do some research uh, your own, and you can get some publications, and if you can enhance the knowledge about it, you can find some opportunities definitely. Because when I was I was in the early days when I was trying to search opportunities, actually no one believes me. uh, like no one really believed me. Like they told me that you won't never find an opportunity related to bioinformatics or cheminformatics. There are no opportunities. That's actually a lie. So there are plenty of opportunities over there. You just need to find, you need to contribute to the open source communities. Open source communities are the main pillar of developing like a new companies, like new ventures related to AI, ML, and bioinformatics, cheminformatics companies, opportunities. So you need to apply, you need to learn, you need to do research, you need to innovate things by yourself. Maybe you want a supervisor. You can also ask from me. I can help you, and you need to apply. That's how you can get opportunities. So here in I showed in the session your senior students Kalana and some students Shakir, they got opportunities. They worked with me. They helped me to develop scientific communities, some open source projects. They got opportunities. They did something different. So you can also do like that. So I'm IT student from Mounia University. Sir, please can give you me a way to contact you, sir, LinkedIn or email. You can get the presentation uh, from the MLS Society. In there, you have my email, all the details. You can contact me, and you can get my personal email from the moderators as well, or I can type it in here. Like maybe you don't have contacts with them. Uh, okay. Uh, Agentic AI and agent AGI come soon. Agentic AI systems. Agentic AI system is already here, right? The agentic AI system is actually what we are currently using in our day-to-day life. Actually, like earlier it was earlier earlier it was that we are just using AI models through chat interface, but now the system is changed into AI agentic systems. That's why you can connect those large language models like ChatGPT with other applications. Like I can show it in you here if you can give me a second. If you go to the ChatGPT, um, you can see a section in here called apps. So you can connect these applications with ChatGPT really easily. So this is what agentic systems do. They automate all the things, not just providing predictions. They can do the prediction, like then they can as an example, if you can get it, like if you want to send an email to some person through an agentic system, if you think the agentic system can do all the things for you. He can write the letter for you. He can search the email that you want to send the email. He can prepare the email for. He can send the email for you. And also, if your email get a reply from that person, he will also send it to you as well. Likewise, the agentic systems are now available, and there are some tools that also you can use to build your own agentic systems, like Na10. LangChain is one of the agentic AI building tool. You can use LangChain to connect all AI models in databases, like I told you earlier. In here, so it's out there right now. Actually, the things are changing rapidly because of the AI. Okay. What else? Thank you very much. Okay. Anyone has any questions? I can provide answers.
Okay, I'm Dr. Buddhi, Mr. Anuttara. Actually, can you hear me?
Yeah, madam. Nice to meet you
Yes,
after some time,
yes.
madam.
Actually, I would like to thank you first for agreeing to do this session to our students. And also, thank you very much for your timely need, most important informations here. Thank you very much, really, and also really proud of you, Mr. Anuttara. Okay, thank you very much.
Thank you, madam. Thank you.
Okay.
Yeah, Anuttara, I am Dr. Kalani here.
Yes, madam.
Yeah, so I actually we were amazed by your very informative talk and all of your achievements, like Dr. Buddhi said. So you have gone so far, learning many things and also open up novel opportunities to many of our students as well.
Yes.
So we are really proud of you, Anuttara.
Thank you, madam.
So, like you said, I think uh, like we would like to build up collaborations with you. It will be very good for our students to think of new pathways.
Yes,
So,
madam.
what
Definitely.
is like your advice? Like you said, it's good to do some self studies
Yes.
for them if they want to develop in this field. Anuttara. So,
Yes.
uh, like should they learn like some coding during their this
Yes.
undergraduate period? But what is your advice for our students, Anuttara? Like if they want to like work in this field, because they need to develop some skills, some
Yes.
computational skills as well, right?
Yes, madam.
Or
Yes. Yes. Definitely, madam.
so
They need to learn. They can learn by themselves. Actually, there are a lot of
Mhm,
sources out there because I learned myself, and I
mhm,
contacted a lot of people and sent like 1,000 emails. Uh,
right.
so that's how I did. But it is better if we can kind of add some curriculum uh, for our courses in the university as a course, uh, like AI, artificial intelligence, and machine learning, and those things, informatics subjects. If we can kind of update our curriculum, it will be better for the students, madam, because
Mm,
we can give that knowledge from the degree level as well.
yeah, yeah. So that's we are very grateful for your insights, Anuttara. And actually, we are honored to have you as the inaugural president of the MLA Society.
Yes.
We are
Thank
very
you.
proud of you. And so, dear students, also now Anuttara is a very good example how he has come all this way doing self studies and self research, and now he is leading a company, giving novel opportunities to many of you. So, you also can, join Anuttara and learn from him, right? Thank you very much, Anuttara.
Thank you, madam.
So,
Thank you,
yeah,
madam.
like the junior students should also know, like Anuttara was the very first person who, helped us in buying the musical instruments and everything to the faculty those days when you were an undergraduate, isn't it,
Yes, that
Anuttara?
was five
He was,
years back, 5 to 6 years
yeah, he was a great bass guitarist. I think nobody touched the bass guitar after
back.
you, Anuttara. You donated the stand also, and it's still sitting there. Yeah. So,
Students need to
yeah,
have their own courage to do things, madam. We
yeah,
can't
yeah,
force them
that's it.
always.
Yeah, you had that courage since that time. So we are really proud of you.
Yes, we
So
can.
thank you very much for this informative speech. Actually, I also learned a lot of things. Um,
So actually, it's
and it's
really
nice.
hard to explain all the things, madam. The time is limited. There are a lot of things to tell. I am hoping to do a workshop for them because they asked from me. I will do it
Yeah, that will
for
be
sure.
very nice actually.
Yes,
Yeah, because I also didn't know about these neural networks and things, although my husband also works with these things, but you
hmm,
nicely explained how you use these things practically to achieve
yes,
like this day-to-day things.
yes.
So thank you very much for your time and effort. It was a very nice talk. We really enjoyed listening to you. Thank you
Okay,
very much,
madam.
Anand.
Thank you, madam. Thank you. Okay.
Hello, Mr. Anuttara. Can you hear me?
Yes, madam. Yes.
Sorry, previously I couldn't complete my question and get clarified everything that I
Yes.
needed to have.
Yes.
Anyhow, I think as the time will not permit, I will contact you. And
No problem.
actually, I'm very much interested about your content because I have worked for wet labs and experiments related to molecular things and
Okay,
the
madam.
uh, drug developing.
Ah, great.
So it was, yeah, it was really nice to hear that you are working in this field. So I will contact you and let's see the possibilities.
Yes,
So
madam.
thanks a lot for your informative talk today. Good
Okay,
luck
madam.
with your, okay.
Thank you, madam. Thank you. Thank you. All right, I think it's time to finish the session, right? Thank you very much for participating for this session. Thank you.
Thank you very much, Mr. Anuttara Gamage, for allocating your valuable time and sharing your insightful knowledge with us. And we truly appreciate your presence and the inspiration you have given us today. And thank you for the insightful questions and the engaging discussion. And before we proceed further, I kindly request everyone to turn on your video so that we can take a group photo to commemorate this moment. Thank you all. As we approach the conclusion of this session, I would like to invite Ms. Pabudi Manarangi, the Secretary of the Ruhuna Medical Laboratory Science Student Society, to deliver the vote of thanks. Pabudi, over to you.
Good evening, everyone. On behalf of Yohana Medical Laboratory Science Student Society, I'm truly honored to wrap up today's incredible, insightful session with heartfelt thank you. First and foremost, I would like to express our deepest gratitude to the Dean, Professor Imendra Kotapola, Faculty of Allied Health Science, University of Ruhuna, and the Head of the Medical Laboratory Science Department, Dr. Eranga Silva. Thank you, dear sir and madam, for your visionary leadership and for the essential support provided to make this webinar a success. Furthermore, I would like to extend our sincere appreciation to our senior editor, Dr. Kalan Jayasekara, and our senior treasurer, Dr. Buddhi Fernando, for your unwavering guidance and constant support. Next, I have to say a very special and heartfelt thank you to our guest speaker, Mr. Anuttara Gamage. So it is a truly inspiring for all of us to see one of our senior reaching such great success in the professional world. We admire the great determination and commitment you have shown in pursuing your field of interest and chasing your dream. And we are deeply grateful that you never forget the place where you began your journey. Your genuine willingness to give back to your university and your juniors is something we truly value and respect. Your advice and constant guidance means a lot to us. Thank you for showing us that there are so many paths than we imagine. We wish you all the very best in your future endeavors. And a huge thank you to our committee members of the society and our moderators for being the strength behind making this webinar a success. Finally, thank you all the participants for your presence. We believe that the valuable insights shared today will serve as a powerful stepping stone for your future careers. Thank you.
Thank you, Pabudi, for your thoughtful words. With that, we come to the end of this session of our webinar series. We sincerely hope that today's discussion has broadened your understanding of the role of artificial intelligence in the future of drug discovery and development.
Thank you all for joining us today. We look forward to welcoming you to our upcoming sessions. Until then, take care and have a wonderful day.